Important point you need to understand is that whenever this H plus is going out, that means the acidity or the pH of the medium remains constant. That means in anomerization process, okay, or the conversion of Fisherman projection into Haworth projection is actually called anomerization. Okay, in anomerization process the pH change never occurs. Okay, now you need to understand now again what happens when we use fructose instead of glucose here. Why I am talking about fructose, why I am talking about fructose? Because fructose is again a very good example of ketose. Ketose means what? Having ketone group. Okay, now listen, how to draw ketose or how to draw a fructose. This is how you draw a fructose. Okay, okay, this is how you draw a fructose. Now listen, what is the difference between fructose and glucose? In fructose, the primary functional group is present in the second position. The primary functional group is present in the second position, and in glucose the primary functional group is present in the first position. Now listen, there lies the problem. Okay, there lies the problem. Listen, what is the problem? Let's discuss. So if I want to convert the Fisherman projection formula or Fisherman formula of this particular compound or fructose into the Haworth projection formula, then the first challenge I will have to overcome is this: how can I arrange this CH2 and this C double bond O, right? Now listen, the answer to this is, now again you will have to consider this as the what? Head. Okay, always remember how to remember which side you will have to consider the head, which side you will have to consider that which is the head and which is the tail. Now listen, whenever we are talking about the first carbon is actually the head, and you will have to pull that head, right? That means again it is lying down here again and again. Okay, I have already told these things. That means at the end what will happen is, listen, there is a trick. What will happen? Now listen, that is the difference. What will happen when we talk about fructose? This is the first carbon, this is the second carbon, this is the third carbon, fourth, fifth, and this is the sixth carbon. Now listen, if I pull this head, then what will happen? Again, this is the second carbon, this is the first carbon, this is the second carbon, this is the third carbon, fourth, fifth, sixth. Okay, now listen with all your attention. This is what? The third carbon. In the third carbon, H is present on the right hand side. That means right. In the fourth carbon of right, fifth carbon, O is downward, H upward. Okay, clear? Don't worry, everything will be same. Now what? That is rotation will occur. Why? Again, because of bulky groups. Here it will go here and it will come down, right? It will come down. Now listen, what is occurring, what is happening is, will there be any space? Okay, I will try. Listen, what is happening here is, OH, right? H, OH, OH, H, OH, H, right? Wrong, obviously wrong. Right? Okay. Now, again what will happen? H plus from solution will attack here. Okay, and lone pair from this OH group, from this oxygen of OH group will attack here. Why? Because this is positive. What will happen? Okay, and this H will be going out as H plus, right? Okay, so this is our what? Structure of fructose, Haworth position formula of fructose. Okay, now listen with all your attention. So what was the difference between the Haworth position formula of glucose and fructose? In the Haworth position formula of glucose, we were getting a 6-membered ring, and in case of fructose we are getting a 5-membered ring, right? This 5-membered ring is called furanose, and that 6-membered ring, which we were getting after the anomerization of what? Fisherman projection, was actually called what? Pyranose. Why they are called pyranose and why they are called furanose? Because these structures resemble a molecule known by the name of furan, which looks like this, and these structures resemble a molecule which is known as pyran, which looks like this. Okay, now we will talk about anomerization or anomers. Okay, clear? Okay, now we will talk about anomers. Now, those who are watching the video right now, you must pause now and you must immediately draw the figure of glucose, that is Haworth position formula of glucose, using the technique which I have told you. Take your time. Now, okay. Now you will have to understand that how can we use this Haworth position formula for our daily lives. It is very important. This Haworth position formula is very important for our daily lives. Okay, now listen. The most important thing you will encounter is this: is what is anomerism? Listen, anomerism occurs only when there is cyclization of glucose. Anomerism only occurs when there is cyclization of glucose. Clear? When there is only cyclization of glucose, then only we will get anomerization. That means whenever Fisherman projection is getting converted into Haworth projection formula, then only you will get anomers. Okay. Now there are basically two types of anomers. Okay. There are basically two types of anomers: alpha anomer and beta anomer. Okay, now how to distinguish between them? Okay, now listen. Don't look at my board. You try to solve it by your own, the Haworth position formula. Okay. Now, when you are not looking at my board, you will get confused. Why? Because this is the first carbon and this is the first carbon. and after that this OH will go down. Okay, now listen, you will have to draw like this, but you will have to come from here to here, that means from top to bottom, and it will go like this. Okay, so clear? Now we know. Sorry. H plus from solution is coming, then it is attacking here, here is the plus charge, here is the lone pair, it is attacking here, so we are getting a good formula, that is getting a good structure which looks like this. Okay, OH, H, OH, H, H, OH, H. Oh. Sorry. Okay, so again nothing has changed. Okay, so we now know how to deal with these idiotic things. Okay, oh. 1 2 3 4 1 2 3 4. Okay, everything is now all right. Now listen. Okay, this is your Haworth position formula. Now listen, if you want to know how will you remember or how will you recognize whether this is an alpha anomer or beta anomer, you just have to remember the first carbon orientation. You will just have to remember the first carbon orientation, that is, in the first carbon whether the OH group is present up or down. If it is present on the top side, then it is beta anomer. If it is present on the downward side, then it is alpha anomer. That's it. Okay, if it is present on the up side, then it is beta anomer, it is alpha anomer. Now listen, so the total thing which will be written in your book is, suppose this is your structure, then it is called beta D. D is actually for stereoisomerism, D and L. D glucopyranose. Why pyranose? Because the pyranose, this particular statement shows that anomerism has occurred. That means the Haworth position formula has been converted into, Fisherman position has been converted into Haworth position formula. That's it. Okay, clear? Now listen. Now listen with all your attention. If I tell you that what is the importance of alpha and beta anomer, what will you say? If I tell you what is the importance of alpha and beta anomer and how we can obtain specifically alpha anomer and how we can obtain specifically beta anomer, are they important? Yes, they are important. Why? listen, if I tell you that we can actually obtain beta anomer, then your question will be why, how, and why we want beta anomers. Listen, let's talk about the how part first, then how we can obtain it. Listen, first thing you need to understand that beta anomer is actually an abnormal anomer. Why it is abnormal? Because if you take a sugar solution and if you heat it above 98 degrees Celsius, then all the water will evaporate and glucose will be left out. Now remember this, all the glucose which are left out in the pan are actually beta anomers. Take my word for it. All the anomers will be present, all the anomers will be beta anomers. And how can we obtain alpha anomer? Take a sugar solution, keep it here for 1 week. All the water will again be gone, but this time due to evaporation, not due to heating, right? Then all the anomers which will be present here will be alpha anomer. That's it. It's so simple. That means what? That means alpha or beta anomer totally depends on whether you are heating the solution above 98 degree or below 98 degree. That's it. Simple. That means what? Beta anomers require extreme conditions for the formation, right? And alpha anomer requires normal room temperature. Okay, right? Now listen. So you might be thinking that in our body, then we don't have 90 degrees Celsius. The inside of our body is actually not 90 degrees Celsius. That means our body contains mostly alpha anomer, right? I can prove you mathematically. Don't worry, I will prove that in the board. I can prove you mathematically that alpha anomers are only present 36%. I can prove it right now. Beta anomer is present at a more percentage. Why? Listen, when you talk about biochemistry, you're actually talking about chemistry, and chemistry actually deals with reason, not with expectations. Life also works like that, but lovers and idiots don't understand that. Now listen, most important thing you need to understand today is this, that which form, that is alpha form or beta form, alpha anomer or beta anomer, which one is stable? Depending on that, which one will be at higher percentage, we will calculate. Now listen, before we go into the details of these discussions, I would like to notice, or I would like to take your attention for telling something very important right now. Listen, we will perform an experiment right now, okay? And for this experiment, we need a very important, machine, okay, which is called X-ray crystallograph. That's it. We need an X-ray crystallograph. Why? Because we will be talking about some angles. Listen, if you don't understand what is X-ray crystallograph, don't worry, we will have a separate class on X-ray crystallography. Okay? Now listen. Suppose this is a jar. Okay? Suppose this is a jar. Please don't mind if the alignments are not right. Okay? I'm not expert in aligning everything. Now listen. Now listen. Suppose this is a jar, this is a jar. You have kept here, I'm telling you, you have kept here alpha D glucopyranose. Okay, you have kept here alpha D glucopyranose. And here you have kept what? Beta D glucopyranose. Now, if you subject alpha D glucopyranose, you will understand, just for your own sake, just remember this. Suppose I am a light, okay. Suppose I am you will have to learn Fourier transformation before you can learn X-ray crystallography. Now listen, for your simple, you need a simple explanation right now, I am giving you one. Listen, suppose I am a light, I am moving, okay, and suppose I am a light, you come here, okay, stay there. Suppose I am light, come here. Is she coming in the camera? Okay, stay, focus the camera. Now listen, suppose I am light, I am coming here, and if after interacting with this particle I am going toward left, then my angle of rotation will be positive. sorry, if I'm going towards right, my angle of rotation will be positive. Suppose she is a particle and I'm light. You know light have particle nature as well as wave nature. Suppose I am following the particle nature right now, and she is a particle. Two particles can interact, and after interacting, suppose I am going towards right, then my angle will be here positive. And if after interacting with a particle, I am going towards left, then my angle will be negative. Okay, thank you. Okay, now listen with all your attention. Suppose I am light and she is the glucose. Okay, don't worry, you don't have to come again. Suppose she is a glucose. Glucose interacts with light like a particle. Now listen with all your attention possible. Whether it is alpha NMR or beta NMR, what are you thinking? Whether it is alpha NMR or beta NMR, listen again. Whether it is alpha NMR or beta NMR, glucose always deviates a light particle positively. Okay, clear? That means whenever, suppose I am a light particle and she was glucose, suppose I don't know whether she is alpha glucose or beta glucose, I will go towards the right. But suppose she is alpha glucose, what will change? I will be going towards the right also, only, then what will change? My angle will change. Why? Because whenever light interacts with alpha D-glucopyranose, then the angle will be 112.2 degree plus. You know plus, why it is plus? That means suppose she is standing there, suppose she is standing here and I am the light particle. If I interact, then the angle of rotation will be what? 112 degrees. That is more than 90 degrees. I will rotate like this. Okay. But if she was a beta D-glucopyranose, if she is a beta D-glucopyranose, okay, then her angle of rotation will be only, you know what, 18.7 degrees, positive. Okay, 18.7 degrees plus 18.7 degree. That's it. Clear? That means whether it is alpha NMR or beta NMR, whether it is alpha NMR or beta NMR, actually both deviates the light positively towards the right. Clear? Now listen. Okay. Now a very important challenge actually was faced by different biochemists who were actually working with glucose. Why glucose was so much important? Because in the early days, you know, scientists understood that our main organ, that is heart and brain, uses glucose. That's why its chemistry was needed, and they started working with this. Now important point you need to understand is this, that whenever I'm talking about alpha D glucose or beta D glucose, you must understand these are interchangeable forms. That means suppose I'm a light and she is standing here. At first, suppose she has rotated me 112 degrees, and suppose another light particle is coming here, she may rotate that another light particle 18.7 degrees. That means what? She is changing. Yes, that's the point you need to understand. She is a very much, you know, tricky person, and she can change her form from alpha to beta or beta to alpha. These are two interchangeable forms. Okay, but there is an important point here again to understand. What is that? Listen. Suppose if I take alpha D-glucopyranose, then the angle of rotation will be 112.2, right? And if I take beta D-glucose, angle will be, right? Now listen with all your attention. If you are thinking these angles will keep the, if these angles won't change, if you think that if these angles are actually constant, then you are actually wrong. Why? Because whenever alpha D-glucopyranose is dissolved in water, they decrease their angle of rotation to 52.7 degree, 52.6. That means it will be degraded to 52.6 degree positive. Okay? But if, now listen, but if you take beta D-glucopyranose here, if you take beta D-glucopyranose here, then this angle will go up to 52.6 degree positive. That means 52.6 degree is actually what? Equilibrium angle, no? Suppose she is standing again here, the particle or sorry, glucose and light particle interacts, then at first she will try to bend my pathway by 112.2. That means what? She's alpha or beta? She's alpha. Now listen, if she is beta, then she will turn my pathway, right? But after 1 minute, what I will see, whether she is alpha or beta, she will turn my angle from up to 52.6 degrees. That means this is the equilibrium. That means whenever alpha D glucopyranose is kept in a solution, at first it will turn towards the right and up to 112.2. Then after that, it will decrease its value to 52.6. And in this case, it was turning 18.7 and it has increased from 18.7 to 52.6. But listen with all your attention. That is the problem you need to understand today. That whenever you are dealing with alpha and beta anomers, you should understand that these anomers change their optical rotation types or optical rotation angles, and it is necessary. Okay, and it is necessary. Now listen, what is the actual form of the glucose when they attain 52.6 degree, right? Listen, there is no actual form. Listen, suppose you have kept here alpha D glucose, and after 1 hour, what will happen? You will get an optical rotation of 52.6 degree positive, right? That means what? That means there is a mixture of alpha and beta now. There is not only alpha here. Okay? Now listen. Suppose you have created a pure mixture of beta D glucopyranose, but here from 18.7 degree where it has risen up to 52.6. That means again here is a mixture of alpha and beta both. Clear? That means some form of alpha always is getting converted into beta, and some form of beta is always getting converted into alpha. Okay, and in many textbook it is written that you cannot tell that how much of alpha is actually getting converted into beta or how much of beta is converted into alpha. Obviously it's wrong. You can calculate it. There are formulas, there are even sums related to this which you will face in your exam, and right now we are talking about that. We will talk about the sums which we will encounter, which you people will encounter. Listen, don't worry, it's an easy sum. Now listen, the question was at equilibrium, the question was at equilibrium, which is the most abundant or which is the more abundant form of glucose, alpha glucose or beta glucose. Okay, question was at equilibrium, which was the most abundant form, which was the more abundant form, sorry, not most, more abundant form, alpha D glucopyranose or beta D glucopyranose. What do you mean by at equilibrium? That means when the mixture was telling us giving us only 52.6 degree rotation. Now listen, how to solve this type of problems. Okay, now listen, it's very important for you to understand that you just calculate two things. Okay, so now we will find out that actual percentage of alpha and beta glucose if the mixture has attained equilibrium. Okay, now listen, for this you will have to consider one thing, that is, just remember, fraction of alpha glucose is A and fraction of beta D glucose is B. Then what will happen? Ball. Then what will happen is, bhalo kore dekh, A into 112.2 degree positive plus B into 18.7 plus degree is equal to 1. Is equal to 52.7 degree positive, right? Now listen. If A plus B, sorry, what do you mean by A plus B? A plus B means total glucose, that is equal to 100%, that is 1. If A plus B is equals to 1, that means 100%, that means what? B is equals to 1 minus A. You are poor in mathematics with respect to these levels also. Okay, put this formula for B in this equation. Okay, you will get A is equals to 36%, 36.25, and B equals to 64%. That means B was the percentage of beta. Beta is present in more concentration. That's why. Now listen. What was the extreme angle of alpha? 112. What is the extreme angle of beta? 18.7. Now listen, 52.6 is more or less near 18.7, no? That's why, because it is present in more concentration. Without knowing even the percentages also, you can calculate which is more. Clear? This was your one of the exam related question. Okay. Now we will be discussing epimers.